Okay, so today we're going to be picking back up with the diffusion and exchange of gases. These first couple of slides we're going to go through are a little bit going to be a review of what we were looking at in pictorial form at the end of Monday. And hopefully we're going to move into gas transport. We'll be talking basically about how oxygen gets carried on hemoglobin and the factors that affect that. And possibly we'll get into how carbon dioxide is moving around in the blood as well. Okay. When we were talking last time, we talked about the fact that gas exchange is happening in the lungs. Right? We breathe in air from the environment. We pay attention to those partial pressures of especially oxygen, but also carbon dioxide as they're changing, mixing with dead air that is in our lungs, that remaining part that we didn't breathe out. Then once we have air in the alveoli, we have a really thin barrier between the air in the alveoli and the blood in those capillaries around the alveoli. So the barrier for diffusion is really small because we just have three-ish layers. Right? We have those type 1 alveolar cells lining the alveoli, so they're part of the lungs. We have basement membrane, and then we have the capillary endothelial cells. We also know that that round shape, those great like clusters of the alveoli, really expands the surface area for this gas exchange to happen, and this close contact between the alveoli and the capillaries also helps us. So we've got a thin membrane, that's the small diffusion barrier, and we've made a lot of modifications for this large surface area to have all this contact between the alveoli and your blood. So we talked a bit about partial pressures in the blood at the end of Monday. So here we see it in a kind of different form, in a graphical form. So what we're looking at are partial pressures of oxygen. And we can see the partial pressure of oxygen of our mixed venous blood is labeled here. So what we're looking at is blood coming to the lungs. So out in your system, different tissues are using different amounts of oxygen. So when we get these partial pressures, we're really talking about mixed venous blood. So this is kind of like an average, because by the time it gets to your heart, it doesn't matter what tissue it was in. So our mixed venous blood has a partial pressure of 40 for oxygen. And actually, by the time we get a third of the length down the capillary that's lining the alveoli, a third of the way, we're already at our partial pressure of oxygen for oxygenated blood. All right, so we're already at 100, so we know that that's going to be our partial pressure for arterial blood. Arterial CO2 already on there. Right. So they actually kind of have like some buffer room here. The capillaries are larger, longer. There's more of them than we actually need in order for all this gas exchange to happen. So this gas exchange happens quite quickly. Same thing is happening with the carbon dioxide. So here we see our carbon dioxide returning to the heart. Or sorry, our carbon dioxide as it comes into the lungs. Right, so that venous blood has a partial pressure of 46 for our carbon dioxide specifically. So that's a little higher than in the arterial blood because it's picked up that carbon dioxide that we're producing as a waste product in actively respiring tissue. So in that tissue that's doing metabolism, producing ATP. And by the time we get down a third of the length of the capillary, again, we're at our arterial partial pressure, this time for CO2, which is 40. Remember, even though we see this number 40 in both the oxygen curve and the CO2 curve, whenever we talk about partial pressures, we are talking about a specific gas. So they're not the same for like any particular reason, it just happens to be true. So these gases are getting exchanged because, like with all sorts of different physical phenomena, chemical phenomena, we have things going down their gradients. In this case, gases diffusing down the partial pressure gradient. So in our respiring tissue, so the tissue that's doing metabolism, creating ATP in order to run its processes, that PO2 in the cells is low, right? So it's low. The blood coming to the tissue is pretty high, right? So the partial pressure of oxygen in the cells is 40 or less, depending on the tissue. So like some, skeletal muscle, right, that's very active when you're on a run might be even lower than 40, right, because you're actively doing something that requires oxygen. This is why when we look at these numbers, we're paying attention to the mixed venous blood. The systemic arteries are at 100 because of the gas exchange happening in the lungs. So our oxygen diffuses from this high partial pressure down its partial pressure gradient, right? So it's going towards the low end, just rolling down that hill. So the oxygen goes into the cells, which is great because we need that oxygen to keep making ATP. So because we've offloaded this oxygen, we've unloaded it. Well, we'll use that word when we look at the hemoglobin. We've unloaded it from the blood. This means our blood no longer has quite as much oxygen, so its partial pressure goes down. So we're still thinking about oxygen here. Our partial pressure of oxygen in the veins, so systemic veins, is 40 millimeters of mercury, so that's our partial pressure. Just note, as you go through these slides, we do really specify systemic veins, and the reason for that, right, is that our pulmonary veins are the ones coming back to the heart with fresh oxygenated blood. So it matters because in that pulmonary circuit, we have kind of the reverse situation going on for veins and arteries and oxygenation. So just a thing to keep an eye on as you're reading questions. Be sure that you know which ones we're talking about. So we have the same sort of thing happening in the carbon dioxide system, our carbon dioxide transport, except our gradient is the reverse. So our respiring tissue is producing a bunch of carbon dioxide, right? Because as we break down glucose, we separate out all those carbons, we get rid of them as CO2, right? That's how we're making the ATP, right? So we're breathing off this CO2. So we create a high partial pressure of CO2 in that respiring tissue. So greater than or equal to 46. So this is our high end of the gradient. Versus in our systemic arteries, so that's the blood coming to the tissue, we have a partial pressure of CO2 of 40. Okay. So as the capillaries move along next to the tissue, these are going to come to equilibrium, right? As we have carbon dioxide travel down its gradient. It's traveling from high to low, which brings up the partial pressure in the systemic veins to 46 at equilibrium with that tissue. So all that's happening here really is we're coming to equilibrium with the air in the alveoli at the lungs. We carry along those partial pressure through our systemic arteries until we get to the tissue. Then we come to equilibrium with the tissue, which means we unload some oxygen and we pick up some carbon dioxide. And then those partial pressures stay the same into the heart and into the pulmonary arteries until we complete the cycle and pick up more oxygen. Pick up more oxygen and offload carbon dioxide. We're just replacing the oxygen that we've used. We're getting rid of the carbon dioxide that we've created, keeping us in homeostasis, basically, keeping those concentrations in the blood. pretty consistent. So in our mixed venous blood, so that blood returning to the heart, the amount of oxygen and carbon dioxide will depend on the metabolic activity of the tissue. So if there's more metabolism happening, there's going to be greater exchange, right? Because all we're doing is coming to equilibrium with whatever the concentrations, right, the partial pressures are in that specific tissue. So if we have a higher partial pressure of CO2, say, in the tissue, the capillaries there are just going to come to equilibrium with that higher partial pressure. They don't have like a rule written down that they can only take up to 46, right? It's all about just coming to equilibrium. So if you were looking at the partial pressure of oxygen and CO2 in any specific vein, right? Like say a named vein in your thigh or something, it might be a little different than these numbers that we're looking at right now, because the numbers we're looking at are basically an average because this blood is all mixed together. Seeing these numbers again and again, right? So these are the numbers that we want to remember, those partial pressures of oxygen and CO2 in our systemic arteries and our systemic veins. So we have some factors that affect the partial pressures in the alveoli, and therefore the partial pressures in our blood, because this is one of the ends where we're coming to equilibrium, right? In the lungs, our blood coming to equilibrium with those partial pressures of the oxygen and carbon dioxide of the air, specifically in the alveoli. So the first thing that makes a difference is our partial pressure of oxygen and CO2 in the inspired air. So like in the air we're breathing in from the atmosphere. So we did those calculations about how we find partial pressures. So we know that gases add up together. So we basically just take the percentage of the gas in the air we're breathing in and multiply that by the pressure overall in the atmosphere. We could change this. For example, if we give someone oxygen, right? Like if we put them on oxygen, what we've done is change the proportion of the air that they're breathing in that is oxygen. I think it's like 95% oxygen when you give someone an oxygen tank. So that is how we change the alveolar partial pressure of oxygen, by changing the concentrations of what we're breathing in. I'm going to skip down to this third bullet point next, because as you note, the last one, the middle one is most important. And this is at the other end of the system, right? So PO2 and PCO2 of inspired air, right, breathing in. This last one is talking about the other end of the system, where we're coming to equilibrium with the tissue. If you change how much ATP you're producing, you're going to be changing how much CO2 you're producing as a byproduct and how much oxygen you're using. So you could change the alveolar partial pressures by changing basically the partial pressures of the gases that are coming back to the lungs. That I don't think we see changing very much. Most critical thing, and the thing we pay most attention to, is kind of the minute alveolar ventilation, right? So the alveolar ventilation, right? When we're thinking about alveolar ventilation, we're thinking about how much fresh air is getting to those alveoli, right? So we know that that has to do with the respiration rate, right? So that was part of our calculation. We calculated how much air the minute ventilation is coming in by calculating the size of your breath, right? Tidal volume about 500, and then we multiplied it by how many times are you breathing in a minute. So we said that the average normal was about 12. Right? And then the alveolar ventilation, we subtracted out the dead air space. So that was the difference there, taking account of the fact that we have air mixing. Okay. So if we change that, then this is also going to change these concentrations. So we're going to see that when we're controlling partial pressures of oxygen and CO2, right, we know that people breathe faster or breathe slower in certain situations, right? So that's kind of what's going on. We change those partial pressures by changing our respiration rate. So some of this is normal, right? So we have this word hyperpnea. Hyperpnea means that we're breathing faster. So we have an increased ventilation. We're breathing in more air per minute because of an increased demand, right? So our body is using a bunch of oxygen. So we need to get it more oxygen. So we need more oxygen coming into the lungs and coming into the blood. So this generally causes relatively minimal changes in the arterial partial pressures of oxygen and CO2 because we're basically just replacing that additional use of oxygen and production of carbon dioxide in the tissues, right? So we're breathing faster, but it's still keeping those partial pressures in the blood the same because we're using up that oxygen. Basically, we're compensating for the fact that we're using more oxygen. When we talk about hypoventilation and hyperventilation, these are going to change our arterial partial pressures. When we say someone is hypoventilating, what we mean is that their ventilation isn't meeting their body's demand. So they're not breathing fast enough in order to keep those partial pressures the same. And therefore, really not enough in order to deliver enough oxygen and carry away enough carbon dioxide. So if we're hypoventilating, right, what we're saying is we're not breathing fast enough to get enough oxygen to the tissue, but the tissue is still using oxygen, right? So overall, we see our arterial partial pressure of oxygen decreasing because we're not breathing enough, and the circulating blood, right, still trying to use that oxygen at the tissue end. For the CO2, right, it's going to go up because basically we're still producing a bunch of CO2, but because we're not breathing fast enough, we're not breathing enough of it out. So that's staying in our blood. When we talk about the renal system next time, we're going to talk about different ways that we can change CO2 concentrations in the blood. And so we'll kind of come back to how breathing relates to these concentrations at that point. So hyperventilation is the opposite. So hyperventilation, right? So you're feeling really panicky, you're breathing really fast. This means your ventilation is exceeding demands. So you're breathing faster than your tissue is actually using the oxygen, faster than we're producing carbon dioxide. So this has the effect of putting more oxygen into your blood. So we're going to see a higher partial pressure of oxygen. We're going to see a lower partial pressure of carbon dioxide, because if you breathe in really fast, you're also breathing out really fast. So breathing rates has a big effect on our partial pressures of both oxygen and carbon dioxide. There are lots of other words we can use to describe basically the effects of breathing, the types of breathing, and the amounts of oxygen we see getting to the tissues, getting in the blood. So these are just a couple of them. I think we've seen at least some of these before, but you'll probably hear these in the future as we start to think about sort of clinical applications of these. Next, we're going to move into transport of gases in the blood. So we're going to be moving from this kind of big picture of what's happening between the lungs and the blood, the tissue in the blood. We're going to start thinking what's happening like inside our red blood cells, in our plasma. What's going on there? How are we moving this oxygen and carbon dioxide? So that's what we mean when we say we're describing transport of these gases. We're talking about like how are they physically traveling through our bloodstream. So hopefully it's not a surprise that when we think about oxygen, we are immediately associating that in our minds with hemoglobin. Right? we looked at hemoglobin. We know it's in our erythrocytes, right, in our red blood cells. And it's really important for oxygen transport, although it does have a little effect on carbon dioxide as well. So oxygen is not very soluble in plasma, right? We saw that its solubility was actually much less than carbon dioxide. We talked about that when we talked about partial pressures in air versus in blood. So it's not very soluble in plasma, but we do really, really want to carry oxygen to our tissue. So it's going to hitch a ride on hemoglobin. About 1.5% of oxygen in arterial blood is in the plasma, so in the water part of your blood. The rest of it is on the hemoglobin, bound hemoglobin. So we have two words that we will use for different types of hemoglobin. By types, I mean hemoglobin that currently has oxygen bound to it versus hemoglobin that's been emptied of the oxygen. So deoxyhemoglobin means what it says. So D removed, oxy for the oxygen, hemoglobin, because we're talking about hemoglobin. So deoxyhemoglobin is just saying that we're talking about a hemoglobin molecule, so it's protein, without any oxygen bound to it. And if you see it in equations, it's going to show up as just HB. Oxyhemoglobin is the one that has oxygen on it, right? So we're going to see that in equations as HB, little dot, and then oxygen telling us that we have the oxygen bound to the hemoglobin. So we have a reversible reaction happening between oxygen and hemoglobin. So we can take our deoxyhemoglobin, our hemoglobin that doesn't have oxygen on it yet. When it comes into contact with oxygen, the oxygen can bind to it, creating our oxyhemoglobin. And we're also going to see at the tissue end, the reverse is going to be happening. We're going to be taking hemoglobin with oxygen bound to it, our oxyhemoglobin. We're going to be separating out that oxygen. We're going to be unloading it onto tissues, leaving deoxyhemoglobin in our venous blood. So we're going to be loading oxygen, we call it at the alveoli. All right, so loading in the lungs creates oxyhemoglobin. And we're going to have unloading at tissue, creating the deoxyhemoglobin. So in these pictures, we're just zooming in on what's happening in each of these locations. So here in A, we're looking at the lungs. So we see that close contact between an alveolus and the capillary. We see we are at a partial pressure of 100 of oxygen. Our blood coming in is at a partial pressure of 40. Oxygen is crossing that thin membrane, right? So our alveolar cells, the basement membrane, and the capillary endothelial cells. It's coming into contact with hemoglobin in the red blood cells, creating our oxyhemoglobin. And at the tissue end in the systemic capillaries, we have the opposite happening. We start out with our oxyhemoglobin in the blood, right? So our arterial blood has oxygen bound to it. We separate out the oxygen, pull it into the tissue, leaving just the hemoglobin empty of oxygen into the venous blood. So this is how we're physically getting that oxygen into the blood. When we say we're coming to equilibrium, this is where we're putting that oxygen, or this is where we're removing the oxygen from. So every hemoglobin molecule can bind up to 4 oxygen. So we have 4 spots. We have those 4 heme groups with the iron in them. So we looked at that structure in our last unit. binding oxygen to hemoglobin follows the law of mass action, which is to say if we have more oxygen, more of it is going to bind to hemoglobin because more of it is around, more of it is coming into contact with the hemoglobin. So we could have no oxygen, we could have just one oxygen. Whoop! Sorry, didn't mean to scream. Scared me. Okay, or we could have up to 4 oxygen molecules in the hemoglobin. So we will be talking about saturation of hemoglobin as we talk about different parts of the system and different effects on the affinity of hemoglobin for oxygen and vice versa. So when we talk about that saturation, we're basically going to be talking about, so like how much of the hemoglobin has all 4. Sometimes we're going to see we're only filling up 3 of these spaces and so on. This one with the 4 oxygen molecules on here is 100% saturated. This one on the left has no oxygen bound to it. So this is what we would see after we unload oxygen in the tissue. So now we have our oxygen and the carrying capacity of blood. So what we're looking at here, just in this diagram to clarify, the big circle is a red blood cell, so that's an erythrocyte. And each of these little like Punnett square looking things is a hemoglobin molecule. So the little purple bits inside are the oxygen. So when we talk about the oxygen carrying capacity of blood, we're saying, okay, how much oxygen could we put in a liter of blood, right? So when hemoglobin is 100% saturated, so when we have all those 4 spots for the oxygen filled up, a gram of hemoglobin is going to contain 1.34 milliliters of oxygen. We do the math out. The normal blood hemoglobin levels are 2 to 17 grams per deciliter. You did this overall math, I believe, in phys lab last semester, but here's the number we're really going to care about, if we care about any of them, is that the oxygen carrying capacity of hemoglobin is about 200 milliliters of oxygen in 1 liter of blood. So that's how much oxygen we could fit in a liter of blood. This is not necessarily how much oxygen we are going to see in any blood if we sample it, though. We're not always at full saturation, right? So that's kind of like our upper limit. In arterial blood, we're almost there. We're 98.5% saturated. So that's what we're looking at here in this top image. Right, we can see almost all the spots are filled on the hemoglobin. So we have a couple of little empty spots where we could carry a little bit more, like right there. In the venous blood, we're only 75% saturated. So we can see most of these have 3 little oxygen bound out of the 4 places. So we have a lower saturation of oxygen in the venous blood, but it's not actually at 0. We're not completely sucking all the oxygen out of the blood, which we know because we know we have a partial pressure of oxygen of 40 in the blood. So here we're looking at a curve comparing how saturated the blood is. So like what percent of those hemoglobin slots are filled, right? Versus a partial pressure of oxygen. So we can do this because remember that this works by the law of mass action, right? So if we have a higher partial pressure of oxygen, we have more oxygen, we're going to have a greater saturation of hemoglobin because more oxygen is coming into contact with the hemoglobin and therefore more is binding to the hemoglobin. So here we have nicely marked our 2 numbers that we want to remember, right? So we know that the partial pressure of oxygen in veins is 40. And we know that that's 75% saturation. And for our systemic arteries, we know that they're at a partial pressure of 100, and they should be at 98.5, so almost 100% saturation, but not quite. So these are numbers we've already seen. But we can see this is not actually a linear relationship, right? There's a curve here, kind of a sigmoidal curve, so almost an S curve. So one part of this S is much bigger. So it's actually true that we have positive cooperativity when we're talking about the affinity of hemoglobin for oxygen. All that means really for us is that once we have like one of the spots on the hemoglobin with an oxygen in it, suddenly the hemoglobin slightly changes shape in such a way that it's better able to soak up more oxygen. So that's why we see this kind of inflection point here. So we see this suddenly more rapid rise in the curve, because we're saying that once we got one oxygen on a hemoglobin molecule, it became easier to load more oxygen onto our hemoglobin. That's the positive cooperativity. The other reason we want to be familiar with this curve, other than just knowing more oxygen in the blood, partial pressures of oxygen, more oxygen on the hemoglobin, is that we're going to talk about shifts to this curve. So we're going to talk about right and left shifts. So when we say that, we're saying like genuinely the whole curve can shift to the right, or the whole curve could shift over to the left. So when we talk about these right and left shifts, we're going to be thinking about them as changes to the affinity of hemoglobin for oxygen. So when we shift this curve, the reason it's shifting basically is because hemoglobin finds it easier, more affinity, or harder, less affinity, to bind that oxygen. So in order to understand that, let's first think about this rightward shift, right? So if we have a right shift, what's happening, right? So here we see, let's think about a point. I find that the easiest way to think about it, right? So say we're trying to get to this 75% line, right? So we're trying to get to a 75% oxygen saturation. We compare our right shifted point to this normal curve. What do we notice about the partial pressure? Did we need more pressure or less pressure to get to that 75% with the right shift? More pressure, right? So we need a higher partial pressure of oxygen to get that same saturation with a right shift. Do you think then that we have higher affinity or lower affinity in this situation? Exactly, yeah. So a right shift is lower affinity for oxygen. And this might sound like a bad thing, but it's actually not, because while it's harder to get oxygen onto the hemoglobin, this means that at the tissue end, it's also easier to get that oxygen off the hemoglobin. So when we think about right shifts, right, we're going to have easier unloading, right? Easier unloading to the tissue. It's a little harder to get oxygen onto the hemoglobin, but we know we have lots of capillaries. We know we're usually filling up our hemoglobin in that first third. So we're not quite so worried that it's hard to get the oxygen onto the hemoglobin. We really care that our tissue is getting oxygen, right? Because the tissue is what's letting our body do all the things that we need to do in our lives. So this is actually going to be, in a lot of situations, quite helpful that we can get more oxygen to the tissue by this easier unloading of oxygen. Okay. So usually in this normal curve, right, our percent of oxygen unloaded from the tissue is just whatever the difference between the saturation of the arterial blood and the venous blood is. Basically make it easier, faster to do that if we have a shift. We're going to be thinking a lot about right shifts, but we could also have left shifts, right? So a left shift would mean that it's easier to get oxygen onto the hemoglobin, so we have a higher affinity for oxygen. But the converse will be true at the tissue end, right? This means it will also be harder to get that oxygen detached, to unstick it from the hemoglobin, so it will make it harder to get it to the tissue. So as we go through the next couple of slides, we're going to be thinking about different things that affect this curve. So different things that either increase the affinity of hemoglobin for oxygen or decrease the affinity of hemoglobin for oxygen. So we're going to be thinking about, okay, what does that mean for partial pressures and saturation? But especially we want to pay attention to what does that mean for the unloading of the tissue? Are we going to find it easy or hard to get oxygen into the cells that need it? So one thing that has an effect is the pH. And overall, we're going to see temperature has effects, pH has effects, partial pressure of carbon dioxide has effects, and an additional molecule that we haven't talked about in the past, but is produced in low oxygen situations, is also going to have an effect on this curve. So we'll go through each of those, but first we're thinking about the pH. So this is called the Bohr effect. So in the Bohr effect, what we're saying is that a lower pH, so lower pH means more hydrogen ions, increases O2 unloading. Increases unloading of oxygen, right? This means that we are going to be able to get oxygen off of the hemoglobin and into the tissues easier. So this is helpful because active tissues, so metabolically active tissues, doing cellular respiration, they're actually producing acid as they are doing that process. Carbon dioxide actually counts as a biological acid, and we'll talk about why that is later. Okay. So active tissues are producing acid, which means that the pH decreases at the tissue end, right? So if tissue is really active, we are making that tissue acidic. You could also associate this with, we know that we can produce lactic acid in tissues that are really working hard, right? So acid at the tissue end means that it becomes easier to unload the oxygen, which is great because those tissues producing the acid are active, meaning they need more oxygen, right? So this is really helpful that this decrease in pH is causing a right shift to the saturation curve. I'll just quick draw those on again, right? So here we had our normal curve. So we're talking about a right shift. Right shift. Okay. So the Bohr effect says we have a right shift, meaning we have easier unloading and a lower affinity of oxygen for hemoglobin. Lower affinity, easier unloading. And we're going to see that a lot really all of these effects, I think, are going to be kind of related to this, right? So related to this idea that we want to respond to tissues that are using oxygen and producing carbon dioxide by making it easier to get oxygen to them, right? Making it easier to unload. So that's why we're going to see a lot of right shifts, basically. The next does relate to carbon dioxide, but we're actually still talking about effects on this oxygen curve. So the presence of carbon dioxide itself actually has an effect on the affinity of hemoglobin for oxygen. So carbon dioxide can bind onto hemoglobin. It doesn't bind at that same site. It doesn't bind at that heme group where the oxygen does, but it can bind on nevertheless. When it does, it produces something called carbaminohemoglobin. Right, so let's go with that. The carb part is the carbon dioxide. The amino part is like amino acids, so that's telling us kind of about the protein. And then hemoglobin, just our hemoglobin molecule. So the carbamino effect is telling us that we're talking about the fact that when we bind on to hemoglobin with our carbon dioxide, we produce carbamino hemoglobin. So carbamino hemoglobin, so the hemoglobin with a carbon dioxide bound onto it, can still bind oxygen, but it has a lower affinity for oxygen than deoxyhemoglobin itself does. So once we put some carbon dioxide onto the hemoglobin, it becomes harder for oxygen to get onto that hemoglobin. Now, this is useful for us because of the effects at that tissue end, right? So our active tissue is producing carbon dioxide as a byproduct, right? We want to breathe off that carbon dioxide. So when we have increases in metabolic activity, we're increasing the CO2 at the tissue. And this is going to work by that process of mass action as well, right? So if we have more CO2, we have more carbaminohemoglobin, right? And when we have more CO2, more carbaminohemoglobin, overall, that carbaminohemoglobin is lowering our affinity for oxygen, meaning it's easier to pull the oxygen off, meaning we can get more oxygen to that tissue. This is another sort of domino effect of actively respiring tissue, right? So we're going to see a lot of ways that tissue increasing its metabolic rate makes it easier for us to get oxygen into that tissue. So the next is the effect of 2,3-DPG or 2,3-BPG on this curve. Don't worry too much about the specific names. This is a lot of words, but we're going to go for like kind of the high view concept here. So basically, your body produces this molecule 2,3-BPG when there are low oxygen environments, right? So this might be that you are anemic, right, and you just don't have enough oxygen because of the effects on the hemoglobin itself. But this can also be because you're in an environment where the air has less oxygen, which specifically happens at high altitudes. If you've thought about, like, ever heard about getting altitude sickness, right? That's because of the change of this mix of gases in the atmosphere as you go high up on a mountain. So in a low oxygen environment, you produce this as a byproduct of metabolism. If there is enough oxygen in the environment, you do metabolism like normal, you produce ATP like normal, then this compound is inhibited because oxyhemoglobin inhibits this process. So you're only going to see a buildup of 2,3-BPG if you are in a low oxygen environment. You're relying a lot on glycolysis because of that lack of oxygen. Okay. So if you're in this low oxygen environment and you have this buildup of 2,3-BPG, This means you decrease the affinity for hemoglobin for oxygen. Right, so we're again just talking about a right shift, like our two previous examples as well. So this might initially seem counterproductive, right? Because we're reacting to a low oxygen concentration by decreasing our affinity for oxygen. But this is helpful because what we actually care about is tissues getting oxygen, right? We want our tissue to get oxygen regardless of how much oxygen we have in the environment. So we decrease the affinity of hemoglobin for oxygen, meaning we can unload the oxygen that we do have to the tissue. So those are three kind of like chemical effects on this curve. We'll talk next time briefly. Temperature also has an effect, but spoiler alert, active tissue, higher temperature, more unloading. Carbon monoxide is kind of a separate thing. It binds in that same spot where oxygen would, so it's not about the curve, it's about taking up that physical space. Okay. So we actually will not meet here on Friday. I'm going to take a look at our schedule and make sure that if we skip the day, we can stay up with the schedule. If not, I'll have a short video for you guys to watch and I'll talk about it when we meet next time. I will see you next on Monday.